My research topic is on investigation of the mechanism of melatonin's effect on amyloid membrane interaction. This research topic is developed from the fact that neuron cell death and dysfunction is an important step in Alzheimer's disease pathogenesis. Amyloid plaques are found in Alzheimer's disease patients' brain, yielding the presence of oligomeric amyloid species, which has high neuron toxicity through nonspecific interaction with the neuronal cell membrane. Any structural change happens to the cellular membrane should have an effect on the amyloid membrane interaction. Melatonin, a small molecule hormone that is naturally occurred in human bodies, was shown from cellular and animal studies to be able to prevent membrane from amyloid toxicity. So we want to reveal the molecular mechanism of such preventive role of melatonin by using several nanotechnology tools to complete this research from many aspects. So the very first method is localized surface plasmon resonance, where it uses gold nanoparticles as substrate and use it as a sensing surface to detect lipid membrane formation and amyloid binding on top of it. Any interaction between amyloid and the supported lipid membrane can be sensed due to the change of local dielectric constant and is recorded as absorbance peak wavelength change from the light source. So if melatonin does anything to prevent amyloid binding, the delta lambda due to amyloid interaction would be smaller. And the second method is called black lipid membrane. This measures electrophysiological property of the lipid bilayer as a function of ionic current. An electrical bias is applied on the opposite sides of the center Teflon partition, where the lipid membrane is formed to cover the hole and prevents ion current. When amyloid interacts with the lipid membrane, it is possible to form ion channels, as shown in this blue box. So this formation of ion channels provides path of ions from one side to the other. Therefore, changes in ion current can be recorded, as shown in these orange boxes. The very last method is atomic force microscopy and Kelvin probe force microscopy. These techniques provide nano and sub-nanoscale resolution of lipid samples. Therefore, the observation of small lipid domains is made possible. The domain structure is an important fact for membrane topologic and electrostatic features and may affect nonspecific interaction between amyloid and lipid membrane. In this case, lipid monolayer is used as a study object. It is formed on a plain mica surface, and due to the natural length difference between different lipid species, they could form domains of different heights and with different contact potential difference. With the incorporation of melatonin, it could result in a change in the domain formation. And this is how the melatonin's effect of topologic and electrostatic feature of the lipid monolayer is recorded. Preliminary results have shown that melatonin has the capability of altering the membrane structure and also affects the amyloid membrane interaction in certain ways.